Hello, I'm Steve Walters, Marine Science teacher from Mission Bay High School in San Diego. And today we're looking at this big ocean out here. The scientists on board the Melville have to look at the water column. The water column goes from the surface all the way down to the bottom of the ocean. And we have 3 scientists on board today that are going to explain the equipment and what it is they're actually looking for. Let's go meet our first scientist.
Hi, I'm Janan Carbush. I'm a second-year graduate student at Scripps Institute of Oceanography, studying chemical oceanography. And this is our baby, the McLean pump. It's a self-contained battery-powered pump that we send down to a particular depth and pump water through the filter, which is in this housing up here, and out through this flow meter right here. So there's a little dial underneath there that tells you how much water flowed through, and we set the timer so that it's down there for about 4 hours and generally pump around 4,000 liters of water for every deployment. So we just retrieved it, so it's been down there for 4 hours, and we're going to offload the data that's contained in the little computer system inside the battery chamber. And then we're going to remove the filters so that they can be frozen in the -80-degree freezer and later analyzed in the lab. We're looking to extract the lipids from the microbes that we collect on the filters. So these are deep sea microbes from 475 as well as higher depths, so like 250 meters. And the end goal is to be able to compare the structures of the lipids that we extract from the microbes to see if there's any variations in the structure based on environmental conditions like anoxia, where there's no oxygen, versus an oxygenated region.
Juan is going to explain to us what it is he's looking for in the water column. Juan is a graduate student at Scripps Institute of Oceanography, and he is using the CDT and the IKMT to find some interesting animals and plankton that comes out from the water column. Go ahead, Juan.
So one of the things you probably heard already is that besides a lot of the phytoplankton and the zooplankton that people are looking in the water column, one of the things that it's also very abundant, it's microbes. So microbes are everywhere. They are probably one of the most abundant type of organisms that you can find in the oceans. They're most abundant than all of the other types like mammals and different types, and there's a high diversity of them. What I'm interested in is looking at this diversity within the water column in this particular basin. So one way of doing this is to take water samples using the CTD and then filter those water samples through different filters that have a certain pore size. So certain type of cells based on the size are getting retained of those filters. And what I'm going to do afterwards is I'm bringing those samples back to Scripps, and in the lab I take those filters and I extract DNA out of it. So the first type of filter that I'm using is this filter here, which is a glass fiber filter, and as you can see it's completely white on both sides. And this filter has a pore size of 1.6 microns. So basically everything that is bigger than that size, which it's a lot of things on the water anyway, like big diatoms and zooplankton and phytoplankton, are going to get retained in this filter. It's going to go through this filter here that is nicely packed in this plastic thing. And the pore size of this filter is 0.2 microns. So almost everything gets retained here. The only thing that is not retained in these filters are viruses. After filtering 30 liters of water this was from a depth of 568 meters, that a sample that we took yesterday from the CDD. So this is how they look afterwards. So you can see that a lot of cells get retained in the filter, and a lot of them have different type of pigments and coloration. So you can see, and probably they're not very happy right now, but if a while ago we should have taken part of this and look at the microscope, maybe in some cases you can see some different cells and different parts.
I'm doing a couple things in the water column. I'm a zooplankton ecologist mainly, and I'm looking for zooplankton. But I also, in this cruise, I'm trying to determine dissolved oxygen in the water column. So I've been taking samples of water, sample waters from the CTD, Rosette, from Niskin bottles, and as you saw earlier, probably, I was preparing these samples to measure the oxygen, and now they're stored, and they're going to be ready for analyzing back in land when we come back to land. So I have my samples ready, and the reason I'm looking for oxygen is because the species that I'm interested in actually diapause in this basin, and I'm going to explain what diapause is. It's actually a phenomena that these copepods undergo every year towards the end of the summer, beginning of the fall, where they reduce their metabolism and create or build storage, lipid storages, and sink deep into the basin to be able to survive the winter conditions. And to be able to find them, I need to know where the levels of oxygen, because even though they are diapausing, they still need oxygen to respire. So I'm interested in knowing where the hypoxic layer or the hypoxic horizon is, because that's where Calanus pacificus, which is the species I'm interested in, is actually located. So after we've filtered the water samples, hola Juan. It's Juan. Okay. So after we filter the water samples, we put them in these little cryovials. So the filters are stored here, properly labeled. And it clicks in here. We put it back in its little tube, and it's ready to be stored. So what we do, we bring this tubing here and put it in liquid nitrogen. Now we have to be careful that we use these gloves, which are pretty thick. So here is the liquid nitrogen. I'm not going to put this in, but I'm going to show you where they are, if I'm lucky. Maybe this. I'm going to put this away here, because I don't need it anymore. We're freezing it to preserve it. So we want to preserve chlorophylls because we don't want them to degrade. We want them to stay as fresh as possible, so when we go back to the lab, we can extract the pigments and analyze the pigments. So we put them in this thing here. I think it's the right one, hopefully, or I'm gonna be in trouble. Okay, no, that's good. So here are the little cryovials, and so they're pretty well stored here. And they're gonna be there until the end of the cruise. As you can see, liquid nitrogen also evaporates, so we have to keep it tight and closed, and so we can have enough liquid nitrogen when we come back to land, since we won't be able to analyze the samples right away. All right, clicks in there, and we can close this. That's good.
All right, we just saw 3 scientists that were showing us different layers or stratification in the ocean. And things that live in the ocean all rely upon the whole ecosystem of the ocean. And tomorrow, we're gonna see what those living things are.